So to continue this lesson, our next topic is genetic engineering. So genetic engineering is the purposeful manipulation of genetic material in order to alter an organism in some way. So with bacteria, if we alter their genetic material, it often will cause them to look or behave differently. So we can alter the genetic material to where they would look differently, such as things like size or shape, or even behave differently, such as learn the ability to produce a different type of protein. A relatively important area of genetic engineering is something called recombinant DNA technology, which involves microbiology. So that is a specific area within genetic engineering that we're going to focus on, as genetic engineering is quite broad and details basically any type of manipulation to DNA. Again, we're just going to focus on recombinant DNA technology. So what is recombinant DNA technology? Recombinant DNA technology is basically taking DNA from one organism and combining it with DNA of another organism. And that combination of the two different types of DNA is known as recombinant DNA. And how this is used in microbiology is we can extract human DNA and insert a specific gene sequence into a bacterial plasmid, so bacterial DNA, and we can manipulate bacteria into producing human proteins. So, for example, some individuals have medical conditions in which they're not able to produce enough or any of a specific protein for themselves, so they have to get supplements or injections of that specific protein. But as far as where that protein comes from, they can actually be made by bacteria now. All we need to do is get the recipe for how to make the specific protein, which is the gene sequence from the DNA, and put that into a bacterial chromosome. And then now that the bacteria have the recipe, they can produce the protein through translation. And we see this happening in this image. Up here we have the gene for some type of specific human protein, and that is being inserted into a bacterial plasmid. So right here we have the recombinant DNA, which is predominantly bacterial DNA, but we do have the gene sequence up here for that specific human protein. And then that gets put into a bacterium, and the bacterial cells do the rest. They grow and divide, they go through replication, transcription, and translation, which ultimately leads them to produce the protein that we can then extract from them and then use for medical purposes. So in order to perform recombinant DNA technology, you do need a specific set of supplies. So one being the DNA of interest. So obviously if your intention is to make a human protein using a bacterial cell, you need the recipe for said protein. So you will need a eukaryotic cell. You will need access to the DNA of the eukaryotic cell. And you don't need the entire chromosome. You only need the region that codes for the specific protein that you're making. So you need a specific gene region. You also need a bacterial plasmid. So if you remember with bacterial cells have their chromosome, which is their non-essential genetic material. And then some bacterial cells get this extra copy of DNA known as a plasmid, which usually has some type of advantageous genetic material in it. So what we're referring to for this is a bacterial plasmid, which is basically just bacterial DNA. Then you need an enzyme called restriction endonuclease. This enzyme is quite important because it is the enzyme that is going to cut the DNA sequences. So I tend to think of restriction endonuclease as like scissors. They cut at specific DNA sequences, so they're used to cut DNA. So if we want to extract a specific gene region from the human genome, we need to cut up the DNA and then pull out only what we need to use. But also, if we're going to be combining that human DNA with the bacterial plasmid, we obviously need to cut open the bacterial plasmid so that we can put the human DNA inside. So that's what's being done here. This is the entire, at least for this example, it's not very big, but this is the entire DNA sequence. Again, we only need the one gene region that codes for the specific protein that we're trying to make. So the restriction endonuclease enzyme would cut that out as far as the human DNA. And that same restriction endonuclease enzyme would cut open this bacterial plasmid here so that the human DNA can fit into the bacterial plasmid. And then obviously if we're combining two different types of DNA from different organisms, there's going to be a gluing process. So the next enzyme, ligase, is an enzyme that can fuse the human DNA with the bacterial DNA to make our complete recombinant DNA plasmid. So ligase would be used as a glue for that fusion. And then all you need is a bacterial cell. So this recombinant DNA plasmid would get absorbed by a bacterial cell. E. coli is the most commonly used because it grows and divides very rapidly. You can get a large amount of bacteria and ultimately a large amount of protein within a relatively short period of time. So when we put the recombinant DNA plasmid into the bacterial cell does the rest. It goes through transcription and ultimately translation or protein synthesis to produce the protein. Does E. coli really understand why it's making this human protein? No, but it has the recipe to do so, so it does it, and then ultimately we can extract it and use it for our purposes. So the mechanism can be seen here. So first we need our DNA of interest. So again, we need to take the entire DNA sequence from the eukaryotic cell, which is going to be an abundance of DNA. And again, we only need one specific region. We just need the gene region for the protein that we're trying to make. So we have all this human DNA, and we just need one little portion. So we need to use the restriction endonuclease enzyme to cut the DNA. And that is going to free the specific gene region that we want to use for this. We are also going to use that same restriction endonuclease enzyme to cut open a bacterial plasmid so that we can fit the human DNA into it. One interesting thing about the restriction endonuclease enzyme is when it cuts DNA, it always cuts it in a way that leaves these single-stranded ends kind of hanging out. These don't have a very technical name, but they're called sticky ends. Remember, DNA is meant to be double-stranded, so if at any point it's single-stranded, it wants to reconnect with its matching nucleotides. So the A's want to be with the T's, and the G's want to be with the C's. So having these little sticky ends in the human DNA and the same sticky ends present in the bacterial plasmid allows the two to reconnect, or to kind of connect and form this recombinant DNA. So the restriction endonuclease enzyme is again used to cut the human DNA and extract the gene, and then cut open the bacterial plasmid so that the human DNA can fit into the bacterial plasmid. Those sticky ends will match up, and then the ligase enzyme is used to fuse everything together. And then at this point right here, we have a recombinant DNA molecule that's part bacteria, part human. It contains a specific human gene sequence for how to make a protein. And we are going to put that into bacterial cells. Those bacterial cells are going to grow and divide, and they are going to go through, again, transcription and translation, and ultimately produce whatever protein was inserted in through the gene, or whatever protein was coded for the gene sequence that was inserted in. So how is this used today? The most common protein that's used via recombinant DNA technology is insulin production. So it used to be, in order to get insulin, we had to extract it from animals, which typically would involve the slaughter of animals. And unfortunately, with the growing rate of type 2 diabetes, obviously that was not a sustainable route. And then because it was animal insulin, a lot of people had adverse reactions to that form of insulin because it wasn't in human form. So ultimately something better had to be done. So they do recombinant DNA technology now. And what they do is they take a human cell and they extract the DNA from the human cell. They use the restriction endonuclease enzyme to cut out the gene for insulin, so that's the recipe for insulin. They use the same restriction endonuclease enzyme to cut open a bacterial plasmid. So then the insulin gene gets put into the bacterial plasmid. Now we have our recombinant DNA down here that is mainly bacterial DNA, but we do have a region here that codes for how to produce the insulin gene from the human DNA. This plasmid is put into bacterial cells. Those bacterial cells grow and divide. They go through transcription and translation, and they produce insulin. And then we can obviously extract that insulin from them and use it medically for diabetics all around the world. So the next two slides are just meant to show you how microbiology can extend into areas where you probably wouldn't think it has any purpose. And one of those areas is forensic science. So one common thing that has to be done in forensic science is matching DNA samples. So if DNA is left behind at a crime scene and you have potential people that you think were involved in the crime, you may have to do DNA sampling to see if their DNA matches the DNA left behind at the crime scene. And that, weirdly enough, does involve microbiology and bacteria. So how this all pulls into microbiology is something called restriction fragment length polymorphisms, which is a mouthful. Basically, restriction fragment length polymorphisms have to do with the way our DNA is cut when it's exposed to a restriction endonuclease enzyme. Remember, the restriction endonuclease enzyme is an enzyme that cuts DNA, but it actually originated from bacteria. Bacteria use it as a defense mechanism to protect themselves from viruses, for example, bacterioviruses that can infect and destroy their cells. But knowing this, if we subject our DNA to a restriction endonuclease enzyme, since we all have different DNA, our DNA would get cut in different fragments or different sizes based on the code and the restriction endonuclease enzyme. So if I expose my DNA to a restriction endonuclease enzyme, it may cut up my DNA into 3 short pieces and 2 big pieces. But if you exposed your DNA to the same restriction endonuclease enzyme, because you have a different DNA code, your segments might be 2 long ones, 1 short one, and 2 more long ones. But it's going to be different. And these differences allow us to match DNA. So the situation here is that there was a crime, and there was blood left behind at the crime. And so that DNA was extracted from the blood, from the white blood cells in the blood, and it was exposed to the restriction endonuclease enzyme, and it was cut up into a series of fragments, and then it was ran through a machine called a gel electrophoresis machine, which separates the fragments based on size, and we got this kind of banding here in the gel. So then we have 2 suspects, Bill and John. So Bill offered to do a cheek swab so we could extract DNA from him, and his DNA was cut with the same restriction endonuclease enzyme, and his fragments were ran through the machine, and his banding produced a pattern such as this, which is quite different from our samples, so we know that couldn't have been Bill's DNA that was left behind at the crime. Unfortunately, when that same procedure was used for John, everything matched up identically, which would only happen if it was the same DNA. So it must have been John's DNA left at the crime scene. So this is just an example of how a bacterial enzyme, the restriction endonuclease enzyme, is being used in forensic science, which is an area that we wouldn't think microbiology really has anything to do with. Another interesting thing that involves microbiology is polymerase chain reactions. PCR is a laboratory technique where we can amplify DNA. So essentially it's how we can make more DNA in vitro outside of a living cell. But it's used in forensics if we need more DNA to run tests on. And it's also used in diagnostics. So we can use it to diagnose if people are infected with a specific, viral or bacterial pathogen, even if they only have a low concentration of it. It's a highly sensitive test. It was used to diagnose COVID infections, and is one that does take a few days to get test results, but works out really well.